Okay, hi, my name is Alex Ramos, and the team is made up by Paola Hernandez and Eric Teran. And our final project is the detection of circulating tumor cells, CTC, through a microfluidic spiral. So let's start with what is. A spiral microfluidic for CTC is a device that uses microfluidic technology to capture and separate circulating tumor cells in the blood of cancer patients. As we can see, we have already a microfluidic spiral chip. Is already elaborated. One of the reasons that we decided to do this break was the interest in how MEMS process can help of fight one of the biggest diseases like it is cancer. And a process like photolithography is one of the most important process in the chip industry. Few industries have this ability and that causes a great demand. So that is why we seek to share this knowledge, having other quality interest approaches such microfluidic, and to expand the skills for others, for other people that is interested.
Now, how does the MEM work? This MEM uses physics and the advantages of microfluidics instead of chemical reactions to determine how many or if there is at all CTC on the bloodstream. This is able to happen by pumping blood in one side of the spiral and making it circulate until it exits through these different lanes that at the end make a tiny pool where you can take each of the samples to further analysis. Each time that the blood circulates through the stream, it makes, like, using centrifugal force, it separates each of the cells. The heaviest cells are going to be pushed back into the outmost part of the microchannel, and in the inner channel are going to remain the smallest of the cells. This can be also adjusted by changing the number of spirals and by changing the force of the pump, of the pressure it's pumped with. As you can see here, at the beginning, it starts everything mixed, and as it goes through the spiral, it separates each of the cells into different groups that are quite defined. Then you can separate those into lanes. After that, you get the analysis just by simply passing the blood through the MEM. This is like from the series of chips that can be called lab in a chip, because you can do the work that one would have to do in a lab, but it's way easier to understand, way easier to carry, way easier and faster to do, and it does the same work way more effectively.
How it helps. This technology is useful for the diagnosis and monitoring of cancer, since it allows the early detection of tumor cells in the blood and the monitoring of the patient's response to treatment. This device may improve doctors' ability to detect and treat cancer before it spreads. Furthermore, this meme is so effective that it can be used in a variety of clinical and research settings.
The advantage of this meme is that it also makes us see the innovation in which MEMS can be applied. There can be many more ways in which microfluidics can be used to further enhance the, how do you say, to enhance the effectiveness of treatment and of exams and tests. With this simple test, you can isolate the cells to tell what type of cancer they come from which type of tumor they are when you analyze them. And also you can check the relative amount of cells that are in a sample of blood to tell how much it progressing the sickness in the body, because the more tumors there are in the body, the more tumor cells you are going to have circulating through the bloodstream. So thanks for listening to our pep talk about the MEM, because it's quite the advancement that something this small and relatively simple can be used for a problem that has been paining our society for a while now. Thank you very much.